This is IMAT 2021, question 32. So here we have a DNA strand which is comprised of this sequence of bases. We're told that a strand of mRNA is transcribed using this sequence, and we have various charts which show, we need to pick which one shows the proportion of bases in the mRNA. So we have DNA, we want to get mRNA. How are these two related? Well, DNA to mRNA, we are finding the complementary bases to each of the bases that are in this template. For example, A is going to match up with U in the mRNA, T is going to match up with A, G with C, and C with G. So if we find out how many bases are in the DNA, we can find the complementary base in the mRNA. So let's count the bases. If we start with A, we can see we've got 1, 2, 3, 4, 5, 6. For T, we've got 1, 2, 3, 4, 5, 6 as well. For G's, we have 1, 2, 3. And for C, we also have 1, 2, 3. There we go. And as we know which bases complement with each other, we can work out the mRNA. So our A value is going to go to U, and our T is going to be transferred to A. And again, with the G's and C's, they're going to switch around as well. So in the end, we end up with 6A, 6U, 3G, and 3C. So what is our chart going to look like? It's going to look something like this, with two smaller sections that are both equal and two larger sections which are both equal, and we can fill these in with our C's and G's being smaller and our U's and A's being the bigger sections. And if we look at this chart and compare it to the answers, we can see that this chart matches answer C.